If it's okay, we resume our lecture. So we will now turn to another type of data, which are epigenetic data, or more specifically DNA methylation data. And not only do we change to different types of data, but we also change to a different type of method. We are no longer using network methods, but rather we're in the realm of supervised machine learning or biomarker discovery. So I will talk about a genomic biomarker of aging, which is known as the epigenetic clock. In my opinion, it's one of the more successful genomic biomarkers that are out there. If we are very honest and look at genomics, then we have to acknowledge that genomic data have not been as successful as we had hoped when it comes to developing biomarkers for various diseases. We don't really have a very accurate genomic biomarker for diabetes or autism or schizophrenia, or most diseases that you can name really haven't benefited that much from functional genomic data. However, there's one arena that has benefited a lot, and that's really aging research. And one reason for it is really this epigenetic clock software. Now, I want to highlight what is meant by this picture. So this is the Vitruvian Man from Leonardo da Vinci, which depicts the perfect physical dimensions of the human body. However, what we didn't have before was really a time axis. We don't know how old should a certain body part be in a 40-year-old, you know. Wouldn't it be nice to say that if we could say the heart tissue should have that age, that would be the proper age, you know. And the epigenetic clock really tries to accomplish that goal of measuring the age of every part of the body. And why is that useful? Because if we can detect strong deviations from chronologic age, it may spell trouble. So for example, if your heart tissue is much older than it should be according to your chronologic age, then maybe you better watch out. But also, conversely, it could be that if one part of your body is much, much younger than it should be, maybe that also indicates that there's trouble. Maybe it could point to cancer or something. So before delving into the epigenetic clock, let me give you a mini overview of other biological clocks. Some of you may have heard of this Hox clock, which is a clock that plays an important role in development, and I won't discuss it at all. You may have heard, just mentioning it, I give you all the clocks that people learn in biology. Then there's the circadian rhythm clock, right? This tells your body, is it morning or evening? But that clock measures hours of the day. It doesn't lend itself for measuring the age of a tissue. Telomere length is perhaps the most famous biomarker of aging. As you know, the telomeres that are the ends of the chromosomes shorten as we get older. A lesser known clock is known as this P16 INK4A. That's a particular tumor suppressor gene, and one can show that the expression levels of that gene change over time, in particular in T cells, not so much in other cell types. And finally is, of course, the epigenetic clock, and its aim is to measure the years of that have passed since birth in certain ways. So in this talk, I will talk about so-called biological aging clocks, which are defined as fairly accurate methods for predicting or measuring the age in years of a subject or biologic sample. And so examples would be things based on telomere length, expression levels, protein levels, and so on. And sometimes people ask me, why do you use the word clock? You know, what do you mean by clock? For me, a method that measures age deserves the label clock if the correlation is very high between the measured age and the true age. And if you want a threshold, let's say the correlation should be bigger than 0.8. Otherwise, it's not really a clock. It's just a biomarker, you know. And so let's evaluate whether telomere length lends itself for defining a very accurate clock. So here I show you a scatter plot from the published literature where the authors relate telomere length on the y-axis to chronologic age. And I chose this study because that was the publication that led to the highest correlations that I could ever find, you know. So in certain ways, I cherry-picked the best study I could find. And what do we see? Look at the panel, the right-hand panel for women. You see that the telomere length on the y-axis has a correlation of minus 0.48 with chronologic age. That's the highest I've ever seen. And let's think about it. Is that a high correlation? So first of all, of course, it's a hugely significant correlation. That's why everyone knows telomere length. If you attach p-values to a correlation of minus 0.48, you get astronomically significant p-values if you have a sufficient sample size. But think of a forensic application. Imagine you know the murderer of a victim has a certain telomere length. Do you think you can infer their age? You know, not really. You know, it's just not accurate enough for that application. Let's turn to another clock. This P16-INK4A is a particular gene, and its gene expression levels on the log scale, which is this middle panel, versus chronologic age, lead to a high correlation of 0.63. That's the highest I've ever seen in a publication. And so in certain ways, that's the state of the art. You know, if you have a biomarker of aging, that's around what people see, a correlation 0.63. But notice one severe limitation. It actually only works in T cells. So it wouldn't be applicable, not even to whole blood. If you have P16-INK4A levels in whole blood, it has a correlation that's close to 0 with chronologic age, let alone for other tissues, you know. So in order then to develop a biomarker of aging that applies to most tissues and that has a greater accuracy, we really need to go to a different types of data. And how about methylation data? Turns out it's been known since the 1960s that as we age, methylation levels change a lot. And I'm just showing you here some recent articles, but just so that you're aware of it, there must be 50 articles that show that methylation levels have a strong correlation with chronologic age. And so what I'm saying then is that when you go to methylation data, you can arrive at what I call disruptive clock technology. You go from correlations between the measured age and chronologic age from 0.63 all the way to 0.96. We get drastically more accurate estimates. And so now I will describe one particular method which was published in Genome Biology in 2013, and really less than 2 years ago, you know. And that clock was developed on data generated by the Illumina methylation array. And I just want to say a few words about it. So the current version of the Illumina methylation array measures the CpG levels, or the methylation levels of dinucleotides, known as CpGs, from 486,000 locations on the DNA. So think about a data analysis challenge, you know, suddenly you have 500,000 variables, you know. In any event, that's the newer version, the 450 array, but there's also an older version which was known as the Infinium 27K array, and it measured about 27,000 CpGs. Now, each CpG, that's a variable. In certain ways, think of it like a probe. And for that variable, you get a number between 0 and 1. Think of it as measuring, in certain ways, the proportion of chromosomes that are methylated at a particular locus, a rough intuition. And so the good thing is there were many publicly available data sets in the Gene Expression Omnibus and other repositories such as Array Express. So here I collected 39 different methylation data sets ranging from whole blood to colon to thyroid, brain, and so on. And notice I collected data from two different platforms, the Illumina 27K array and the 450K array. Now, many of these data sets were collected in order to find disease-related genes, but I ignored disease status in my analyses because it often had a negligible effect on the methylation levels of the CpGs, certainly negligible compared to the effect of chronologic age. But the one thing I need to highlight is I removed all data sets that relate to cancer. Cancer tissue is so different from normal tissue that it would mess up the whole analysis. I also then collected a separate batch of data sets, again from different tissues and platforms, as test data. Right? The first data set is used to develop a biomarker, and the second set is used to test that biomarker.
I have a question. For example, for this one of the modules, Salmon 4, how many genes inside for this specific data set? Like, I mean, not a specific question, but...
Yeah. So I just want to make clear, so we don't have any modules here. Okay, but I have colors, and it's a good question. So what happens is I have all these data sets, and just to keep track of where the samples come from, I color the samples. So in certain ways, they are human beings, and they are colored blue if they come from data set number 2. That's the logic. Yeah, please forget anything you've learned about networks and modules. We are here in the realm of supervised machine learning tasks. You know, I'll give you more details later. You know, but just to give you to answer your question more directly, the number of samples is one of these columns. You know, it will say in one data set there's 43 samples, in another there may be 50, and so on. You know. All right. So the first 39 data sets were used to construct an age predictor, you know. So think of a regression model. We have age and we want to predict it. And the remaining data sets were then used to validate this age predictor. And now the thing is, when it comes to these two Illumina array platforms, they actually only share 26,000 CpGs in common. And I wanted to use, of course, data sets from both platforms, so I restricted my attention to these 26,000 CpGs. But more than that, I removed CpGs that had a lot of missing data, you know. And so in the end, I actually only analyzed 21,000 CpGs. I mean, only is relative. It's a small subset of 500,000 variables, but on the other level, if you construct a multivariate predictor, you may think that, oh, 21,000 is more than enough, you know. In any event, the statistical goal then is to develop an age prediction method based on these markers, and think of it as a regression model. There is a dependent variable, the y, and that will be chronologic age in years. And then there are a lot of covariates, 21,000 covariates. And I mentioned that it's a transformed version of age because you may know when you do a regression model, sometimes people do a log transformation or square root transformation and so on. So I had my own little homegrown transformation that I can show you later, but I transformed it. Why? Because then it fit the data better in the training data, you know. Remember, when you have a regression model, you get model fit statistics, and they tell you you better transform the dependent variable. All right. Now, why can't we just use multivariate regression? Well, the answer is we have too many covariates. And therefore, I chose what is known as a penalized regression model, which think of it as a variable selection tool. It automatically selects covariates out of these 21,000. And there are many penalized regression models. You may have heard of ridge regression, you may have heard of lasso, but there's a third one which can be considered a hybrid between ridge and lasso, and that's known as elastic net regression. That was developed by people at Stanford. For example, boy, I always forget his name. one author was Hasty and Tipshirani, I think. And do I get that right? I hope somebody knows that. Okay, thanks. And all right, so
I have another question.
Another question, yes?
There are many publicly available data, so this is the immunization data. So sequencing that will be different between the all.
Yes.
So how to normalize the? Because I think
Yes.
sequencing that is one of the big factors.
Yeah, so the question is, the data were all generated by different labs, different tissues, different storage conditions, different ways of processing the DNA. And so in principle, the challenge is insurmountable. And when we teach classes in bioinformatics, we would tell the students, you can't just combine it, that's insane. You will have batch effects that will kill you, right? However, there is one thing that allowed me to get around it. No, there were two things. First of all, everybody followed the Illumina protocol fairly well, you know. So Illumina has a certain pipeline they published, and it's my impression that people really followed it. The second thing is DNA methylation data are just more stable. Messenger RNA changes a lot, and for whatever reason, DNA measures, in my opinion, are much more reliable. So there's less inherent variation. The third thing that helped me was that my outcome of interest, chronologic age, has a very strong relationship with methylation levels. Right? Some of you are so unfortunate to study a very complex phenotype, schizophrenia, and then you relate it to expression data. And boy, are you happy if you see a correlation of 0.15, you know? That's almost a Nature publication, you know, if it validates, you know. The problem, I have correlations 0.6, 0.7, totally different level of signal, you know. So the biologic signal is much stronger, you know. And what's the fourth ingredient that saved me? Sample size. I have 4,000 subjects in the training data. So yes, there will be some data that have a huge chip effect or batch effect, but once you have 4,000 samples, it averages out, you know. And yeah, and also, of course, I developed some tools to make these data comparable, you know. So as part of the software, there's literally an algorithm that normalizes your data to my data. So when you upload your data to my web page, there's a button called Normalize Data. And that normalization does nothing else but to align your data so that they match my training data, you know. That's a challenge, but it could be overcome, as I will show you shortly. So then, when I related this chronologic age variable to the CPGs using elastic net, then I arrived at 353 CPGs. So out of 21,000 CPGs, the mathematical algorithm decided that all you need is these 353, you know. And so I will refer to these CPGs as clock CPGs. And let me now show you the accuracy of this resulting predictor of age in the training data. So in certain ways, the most important panel is the upper left panel, which shows you how chronologic age, which is on the y-axis, relates to the predicted age, which is on the x-axis. And what do we see? First of all, there's a very strong correlation, 0.97, And the second, and also notice now the colors, right? The samples are colored by data set. And several comments. Notice the y-axis. Actually, the values go from negative values, which correspond to prenatal samples, all the way to, I think, 102. I have a huge age spectrum. And because I have this huge age spectrum, I observe a very high correlation of 0.97. Imagine if I had restricted attention to middle-aged people, people who are older than 40. Clearly, the correlation would be much worse. I think more 0.6 or so, All right, that's one comment. The second comment is you see lots of panels, and they correspond to different tissues. So, for example, there's in the middle, there's a panel age, and that shows how the predicted age relates to chronologic age in sorted neuronal cells and glial cells. Now, what you notice is in every single tissue do I observe a very strong correlation between predicted value and observed value. And of course, you will be aware that this may be due to overfitting, right? Because these were the data used to train the model. To arrive at an unbiased evaluation, we truly need independent data, and here they are. So these are now the test data for evaluating the test. And please look again at panel A, upper left corner. Here we observe a correlation of 0.96 between predicted value and chronologic age. And that is really my claim then. If you have samples that range from age 0 to, let's say, 100, I claim that my age predictor leads to correlations that are above 0.9. It's a bold claim, and after I published it, I didn't sleep well for several weeks, because it's such a high correlation that anybody who has any experience with supervised learning methods applied to genomic data, they will simply not believe it. It's too good to be true. But in any event, let's take a look at individual panels. So panel C, meaning the third panel from the left, shows you the results for sorted blood cells, so CD4 T cells and CD14 monocytes. So the predictor works in sorted blood cells. It also works, of course, in more complex tissues, from lung tissue to fat, heart tissue, and so on. There's one panel where the prediction is really off. You notice for female breast tissue sample, there's a systematic deviation which indicates that the predictor overestimates the age. And there's really two ways of looking at that result. One interpretation is that the predictor is simply inaccurate in female breast tissue, and it should be modified. So arguably, if I just included more female breast samples in the training data, maybe I arrive at a more accurate predictor. That's one. Yes, go ahead.
Black line is predictor.
Yeah, so the black line indicates perfect agreement. So black line is y equal x, right? And what you see is that the dots are to the right-hand side, meaning the methylation age overshoots the chronologic age. Yes. And coming to the interpretation, but there's a possibly more exciting biologic interpretation, which is, well, maybe the predictor is accurate, but what is different is that the breast tissue is older, Maybe for one reason or another, female breast tissue ages faster than the rest of the body, And that is, at this point, a hypothesis, nothing else, But it gives you a sense of how this type of age predictor possibly lends itself then for measuring biologic tissue age, I do want to highlight that the predictor is very accurate in brain tissue. So all of these are different brain data sets, And as an overall very high accuracy, but even in sorted neurons and in glial cells do we observe a high accuracy. And why is that exciting? Because, as you know, neurons no longer divide. And therefore, we immediately know that the epigenetic clock does not measure the number of mitoses. In other words, it doesn't measure what is known as mitotic age. I mean, many people would think, oh yeah, well, somehow it relates to how often a cell divides, but that argument tells us that's not the case. It really measures a totally different, unknown biologic process. I also want to briefly mention that in this data set, in the lower two panels on the right, I had multiple brain regions available from the same subjects. And therefore, I was able to compare the methylation age of the temporal cortex to that of pons, frontal cortex, and cerebellum in several data sets. And in these subjects, I see no difference in the ages of the brain regions. In other words, all parts of your brain have the same age, with the one caveat that these are middle-aged people. They're in their 40s. Because in a few minutes, I will show you that this result no longer holds in centenarians. When you have the brain of a centenarian, some parts are older than other parts. We'll get to that. Yes?
Has this data been adjusted for some form of genetic makeup?
For what?
Some form of ancestry adjustments or haplotype adjustments?
So it hasn't been adjusted for ancestry information at all. So is that what you were asking? So no. I ignored ethnicity, certainly. I didn't use genetic markers yet, but in a second I'll show you, or I'll tell you a little bit about genetic studies, in certain ways, which SNPs relate to epigenetic age. That's actually a very good question to ask. Other questions at this point? Or? Let me proceed. I'm sure you will have plenty in a few minutes. Yeah? So coming back then to telomere length. As you know, we've worked for decades, we as a field have worked for decades on studying whether telomere length relates to aging. Does the shortening of telomere length explain why we age? And here we had data from the Framingham Heart Study, and Brian Chen analyzed the very same subjects. Every dot here is a human subject. The upper panel on the left shows you telomere length on the y-axis versus chronologic age on the x-axis. And what do we observe? Correlation minus 0.27. For the very same women, we also knew they had not just women and men, we also had methylation data, and you notice we get a correlation of 0.81 in the lower panel, Now, if you had to decide which of these biologic variable relates to the cause of aging, you could revisit what is known as the Bradford-Hill criteria. These are criteria developed in epidemiology for deciding what variables are likely to be causal. And criterion one is strength of the association, So if everything being equal, if this was 1980, we should really focus on studying epigenetics in relationship to aging, as opposed to telomere length, according to criterion one. I do want to highlight, though, there's telomeres as a biomarker have one major advantage over the epigenetic clock, which is we understand the biologic mechanism. We really understand why telomeres shorten. We understand why they are there. We really have a beautiful understanding of the mechanism. In contrast, for the epigenetic clock, we actually don't know which biologic process explains why it is so accurate, So bear that in mind. Let me come to another application of the epigenetic clock. So I was able to download freely available data from chimpanzees and other primates. And when you look at the panel C, the upper right-hand panel, I show you how the methylation age in the blood of chimpanzees relates to the chronologic age, and we observe a correlation of 0.9. Other panels in the upper row show you the relationship between the estimated age and age for other tissues, such as kidney and liver. Overall, these data indicate then that the epigenetic clock also applies to chimpanzees, And that's biologically exciting because often things that are conserved or properties that are conserved between two species are biologically meaningful, Conserved processes often serve a purpose. I briefly mentioned I had some data from gorillas, and there the relationship leads to a correlation of 0.5, but it seems to break down. So keep in mind the limitation here. We use a human Illumina array, and certainly the human array is arguably not applicable to primates that are far removed from us. Some of you study stem cells, or iPS cells, and so you can ask the question, what is the epigenetic age of a stem cell? And the answer is shown in these bar plots. More often than not, I get a negative estimate, or an estimate close to 0. And that, of course, indicates that stem cells are perfectly young. And so, arguably, then, we have already a method for rejuvenating us, If you want to lower your epigenetic age, just simply turn every cell in your body into an iPS cell and you'll be fine, So, but of course we need less drastic interventions, So another thing you may have heard of is the so-called Hayflick limit. As you continue to split cells, and they reach a limit. After a certain number of cell divisions, the cell becomes senescent. And so here I show you how cell passaging, which is the y-axis, how cell passage number relates to epigenetic age in iPS cells and ES cells and so on. And we do observe correlations around 0.3. So cell passaging does affect epigenetic age, but not severely, It's not a correlation 0.9. And so again, as I suggested, this indicates that mitotic age is not the same as epigenetic age. And let me also mention that overall epigenetic age is not another marker of cellular senescence. I have several lines of evidence that suggest that cellular senescence, that is measured by various other markers, really is a different process than epigenetic age. For example, radiation experiments that lead to cells to become senescent don't seem to affect epigenetic age. These are unpublished data, but that's my impression. When we talk about aging, all of us think of an 80-year-old perhaps, but you can ask, does the clock also apply to prenatal sample? And here Jonathan Mill evaluated 179 prenatal brain samples and then related the predicted age to chronologic age. And here the x-axis shows the actual age, and you notice the values look weird, negative, right? Negative means prenatal sample, and minus 0.5. Why? Because everything is in the units of years, And so what do we see? The predicted age on the y-axis is negative, which is nice. The clock recognizes these are prenatal samples. But more than that, we do observe a weak correlation that is actually significant. Correlation 0.15, p-value marginally significant, So arguably the clock even applies to development. I do want to mention that one obvious application is to study the epigenetic age of, for example, cord blood samples and relate it to gestational week. And we did that in small data sets, and so far I couldn't find a relationship in blood, So who knows why that is, But just take it with a grain of salt. But it certainly works in brain tissue. So criticisms of the clock or questions. So some people have criticized the clock because its high accuracy seems to entail that it cannot measure biologic age. And what is meant by that? Imagine I gave you the perfect age predictor, no error. It always gives the age, the perfect age estimate. Then arguably that would be an uninteresting biomarker. And why is that? Because no disease could perturb it, right? See, what we want to have is a biomarker that relates to age, but that also relates to various diseases of interest. Hopefully it relates to diabetes or Alzheimer's disease or various age-related diseases. And so if you have a perfect predictor, by definition, it won't work, you know. So in any event, but personally, I don't think that's our problem here, you know, because as I indicated a minute ago, if you apply the epigenetic clock to older subjects, for example, the people from the Framingham study, you know, we got a correlation 0.8, you know, or when you restrict the epigenetic clock to people in their 60s, you get a correlation 0.4. So I think that criticism is not pertinent. As a matter of fact, I'm a little bit peeved by it, you know, because as a data analyst, you strive for an accurate predictor. Finally, you give people an accurate predictor, and then they criticize you that it's too accurate, you know. That's like the hell of a statistician, you know. So, but in any event, there's another criticism, and that is valid. We don't really know the underlying biologic mechanism, you know. So how? In certain ways, you can say, why do I measure? Why do I use a tool if I don't really understand its inner workings? You know. Having said this, medicine is full of examples of various pills that really are useful, but we have no idea why they are useful. You know, and that's a little bit where we are at with the epigenetic clock. So. But in any event, I do want to mention that by now several people work on using gene expression data, GWAS data, and perturbation experiments to understand what relates to epigenetic age. And for example, Ake Lu, who sits here, has worked on a meta-analysis of GWAS data to identify which SNPs relate to epigenetic age. Or you can use transcriptomic data. Certainly WGCNA would be a tool for finding modules that relate to epigenetic age. You know. In any event, let me give you a few applications just for the sake of illustrating that this biomarker has actually some utility. And I will start out with an application to obesity. So here, take a look at the background of this graph, and you will see hepatocytes, liver cells, in the left panel of a lean person and the right panel of an obese person. In the obese person, you see these inclusions of fat, you know. And it just turns out that the liver samples of obese people is epigenetically speaking older than it should be, and I'll show you that. I briefly want to mention that was a collaboration with Jochen Hampe from the University of Dresden in Germany and other people, and also Tim Spector, Jordana Bell, and from the Twins UK study. In any event, here are the data. The first row shows you how chronologic age on the x-axis relates to the predicted age, what I call methylation age, in different tissues: liver, adipose tissue, muscle tissue, and blood. And again, we observe fairly high correlations, which validate the accuracy of the clock. But also notice that in these scatter plots, I depict a regression line. And what you can do then is, when you have a regression line, you can define residuals. And I will use these residuals as measure of age acceleration. Meaning, if a point lies above the line, then this liver sample is older than expected. If a point lies below the line, the residual is a negative value and the sample is younger than expected. And then I this resulting measure of age acceleration is shown in the lower row. So the y-axis shows age acceleration, and the x-axis shows body mass index. And a quick look at these scatter plots tells you that body mass index actually has a very, in my opinion, a strong correlation with age acceleration. Correlation 0.4, you know. Two comments. First of all, I really believe it because I've validated it in a couple of data sets, you know. But the other comment is, notice there are people whose BMI ranges from less than 20 all the way to 70, you know. That's a really heavy person, you know. So to get this high correlation, you need a big spread. Interestingly, in blood, we don't see such a relationship. And by now we have other data where we've shown that the correlation between body mass index and age acceleration in blood is only 0.09. That's the correlation coefficient, very weak. You really need thousands of samples to see it. Yes?
Who's the blue or the red?
Oh, that's gender, I think.
Okay.
Yeah, so that's right.
Obese versus not obese, and then the data.
No, gender,
Okay.
yeah.
Which methylation chip was used here? Was it the larger chip?
Yeah, 450K array.
Did you find any differences with using a larger chip versus what you had?
No, so the question is, in certain ways, does the epigenetic clock apply to both chips? And the answer is yes. You can upload your old data, you can upload your new data, and the coefficient values don't change. It's the same published coefficient values. The coefficient values from the regression model don't change by tissue, by gender, by anything. They're always fixed, you know. Other question? Yeah.
But like, I guess you wouldn't expect to see differences between the old or new RNA-seq data because both of them are using the same 300
Yes.
CPGs.
Yes. Well, so why don't I see a difference between the two chip types? Fundamentally, because I used both in the training data. I trained my model so that it would, there are some CPGs in my model whose only purpose is to detect, did the user upload the 450K or the 27K? So they automatically adjust for it, you know. And while I'm at it, let me mention that many people think that the 353 CPGs are highly correlated with age individually, right? And that's simply not the case. Don't think that these 353 CPGs have a high correlation with age in your data. There will be some that have a high correlation, but think more like 50 of them, you know. And why is that? Because we have a multivariate model, right? When you select genes using a multivariate model, how does it work? It first takes the one gene that has the strongest correlation with age, chooses it. But what about the second gene? Will it now pick another gene with a high correlation with age? Probably not, because it already has that information, right? A multivariate model will only use covariates that add additional information, right? And so many of these 353 genes have a correlation close to 0 with age. Their purpose is to detect what tissue you're dealing with or other properties, you know. All right, so in any event, that was the finding. So some people are depressed by that. So obesity increases the age of the liver. And then a natural question is, well, if you lose weight, will it restore the age? And so for some of these subjects, we had 2 liver samples that were separated in time by 9 months, before bariatric surgery and after bariatric surgery. And so let me show you the results. So does bariatric surgery affect the body mass index? Certainly. That's the upper panel. It really lowered BMI. But this middle panel shows you the change in methylation age, and there's really no change, which is an unfortunate finding in certain ways. However, so it would indicate that losing weight doesn't restore youthfulness of the liver. But remember one huge caveat, which is that's within 9 months. You know, obviously there's a good chance that the liver will restore itself if you just followed these subjects 2 or 3 years, you know. So take it as with a grain of salt. Yes?
The last one is also use the same model?
Okay, so the last panel is from a totally different study. That's no longer liver tissue, but adipose tissue. And there the authors had evaluated the effect of a 6-month physical exercise intervention, right? And so they had human adipose tissue. And I just threw it in because wouldn't it be nice if we could say if you exercise a lot, your fat tissue becomes younger, you know? I don't have a good reason. It wasn't I just did it because it's a cute little analysis. And
It depends on the exercise.
it depends. So one comment was maybe this finding depends on the exercise. My claim is it depends on the tissue. My caveat, you know. Why would fat tissue show an effect? Maybe if you look at lung tissue, maybe heart tissue or other tissues show a rejuvenating effect of exercise. Why fat, you know? And let me go back this slide. All of us know that excessive weight is not great. But often people confuse the two notions. Things that are bad for you versus things that age you, you know? Just because something is bad for you, for example, you will die faster, doesn't mean that it will affect the tissue age, you know? Example would be BMI versus blood, you know, the effect is very weak. And while I'm at it, let me mention that smoking also doesn't seem to increase the age of blood tissue, you know. I mean, it would be wonderful if blood was a surrogate tissue that captures the adverse effect of smoking, but it doesn't seem to be the case, you know. Now, it doesn't mean that smoking doesn't age another tissue. Maybe if we looked at heart tissue or lung epithelium, maybe there do we observe that smoking ages the cells, just not in blood, you know. Yes?
Has this been looked at in diseases of progressive aging, like progeria, for example?
Yeah, the question is, have we looked at progeria, for example? And the short answer is not, unfortunately not. If you know somebody who has relevant tissues, I would love to analyze it, you know. But again, in progeria, you may know that only certain types of cells are actually affected. And so maybe by studying blood, we don't capture it. Ideally, we would want to have really 30 different tissue samples, post-mortem tissue samples from progeria subjects and controls. You know. Okay. All right, let me move on. I do want to briefly mention, so we have epigenetic age, acceleration, and we also have gene expression data from the very same subjects. And then you can ask which gene expression or which transcripts correlate with epigenetic age. And the answer turns out to be mitochondrial genes have a strong relation, meaning when you have a tissue that is younger than expected, then the nuclear mitochondrial genes tend to be overexpressed, you know. And so I'll invite you to read the paper for more details on that. I want to now come to another application, Down syndrome. That's a collaboration with Claudio Franceschi, Paolo Garagnani, and many other Italian researchers. So you may know that many clinical manifestations of Down syndrome really present almost like accelerated aging. They seem to. So some people think of Down syndrome as what is known as segmental progeria. And we analyzed data from brain and blood tissue, and we found that sure enough, trisomy 21 subjects are older than expected. And here are the data. So here I show you the methylation age in the left panel in blood versus chronologic age. The red dots indicate Down syndrome subjects, and you notice the red dots are always above the black dots. And that means that the methylation age is older than expected. And a formal statistical test shows that as well. So we see that in 2 blood data sets, and we see it in 1 brain data set. However, in buccal epithelium, I couldn't really replicate the result, which might reflect that the buccal epithelial cells was really underpowered. We had simply too few subjects. Let me move to another application, and that is the following. So I was interested to identify tissues that age more slowly. And so we looked at centenarians and supercentenarians. And to give you the punchline, it turned out the cerebellum, which is of course a part of the brain, appeared to be younger than other parts of the body. And that was a collaboration with the late Steve Coles and also with Akelu and Wei Ma and others. So let me describe the study. So we had access to 30 anatomical sites from a 112-year-old woman. Really all sites that I could think of, you know. And for each of these sites, we generated at least 2 replicate measurements. And here I show you, the upper panel shows you how the age acceleration on the y-axis relates to the different anatomical regions on the x-axis. And what do we observe? The leftmost bar, colored in turquoise, corresponds to cerebellar samples. So they exhibit a very strong negative age acceleration. All the colored bars correspond to brain regions. The gray bars correspond to the other regions. On the right-hand side, what we see is bone and blood. So in some shapes or form, it seems to be that bone or blood ages faster than these brain regions. Now, you could criticize the study of saying, well, you have only a single person here, a single supercentenarian. But it turns out the lower panel shows you analogous results for 5 additional centenarians, but only brain regions. And every time we observe that the turquoise bar, the cerebellum, has the youngest age. So I'm very confident then in this finding that in very old people, the cerebellum is younger than other parts of the brain. And just to mention it, here I show you how the methylation age of bone relates to chronologic age, and it high correlation, 0.88, right? Why do I do it? Because in the earlier panel, I showed you that bone tissue was much older than other tissues, and you might then suspect, well, maybe the clock doesn't work for bone tissue. But the truth is it does work even in bone tissue, you know. And so it's interesting that the epigenetic clock even works in tissues that were not used to construct the clock. Example, some people from Zymo research applied the clock to DNA extracted from urine sample, and it really predicts the age. So you can give me a cup of urine and I tell you how old you are. You know, that's before you leave today, please, you know. Okay, so let me mention, coming back to the finding that the cerebellum ages more slowly than other parts. So then the natural question is, well, why does it age more slowly? And one way to look at this is to use transcriptomic data and to ask which gene transcripts are overexpressed in the cerebellum compared to other brain regions. And we find lots of gene categories listed here, nuclear lumen and several other categories, but there's a problem. We now compare different brain regions. Cellular heterogeneity really messes up that comparison. It almost renders it meaningless, you know. So in order to get a sense of which of these gene categories might be causal, we use genetics. And so here Akelu used GBOS data to answer which of these gene categories is enriched for GBOS hits that were found in a study where she related epigenetic age to SNPs. You know, do you see it's a roundabout genetic argument to get at causality, and the answer is this dead-like helicase, RNA helicase activity that seemed to be significantly enriched with these GBOS hits, for example, And I let you dwell on it, what it all means, So let me come to another application. That's an application that involves children who seem to evade aging. You may have seen that on television. There are some children who never grow up. They seem to be eternal toddlers. And that's a collaboration with Richard Walker and others. So here I show you photographs of a child. This is a 5-year-old, and the lower right panel shows you the child at age 15. This child always looked like a toddler, Never learned to speak, never learned to walk. Passed away at age 21 from a lung-related complication. So some people claim then that in principle these children are immortal, They don't grow up, arguably they don't age. Unfortunately, this poor subject, of course, passed away from a respiratory complication. In any event, here is another subject. This child never developed. Even at age 6, she looks pretty much the same age as at age 2. And so we were interested in studying whether the blood tissue of these subjects truly shows that they don't age, or do they age? And so Richard Walker managed to identify a total of 7 children. 5 of them have this disorder. I call it pure syndrome X. They really, so why do we call it syndrome X? Because we don't know the cause. There was not a single genetic problem that we could identify, or hormonal problem that explains that condition. But there were 2 additional subjects. One had Turner syndrome, but again, this child looked like she never aged. And there was a Down syndrome subject who also didn't seem to age. In any event, here are the results. The y-axis shows methylation age, the x-axis chronologic age, and dots are colored by disease status. First of all, the left panel shows how accurate the epigenetic clock is in these young subjects. And that reminds me to make a point. The clock is more accurate in young subjects. If you have teenagers or people younger than 40, it will be very accurate. In older subjects, you lose accuracy. But in any event, the right panel shows the clock applied to subjects who are younger than 12. The red triangles show these pure syndrome X children. And what do we see? We don't see any pattern, It's not like the triangles are all underneath the line, So according to the epigenetic clock, then these children do age, or let me be more precise, the blood of these children ages, But now we would, of course, like to see whether other tissues show a similar trend, Hopefully we find that other tissues don't age, What is this blue sample? Remember, there was one subject who had Down syndrome and also syndrome X characteristics. Sure enough, that person was much older than expected, which again validates our finding that we observed for Down syndrome. Do you remember Down syndrome subjects are older than expected? Yeah. All right. So let me come to yet another application, which is to compare the offspring of centenarians to age-matched controls. And why do we do that? So imagine you want to understand what is special about centenarians. Why do they live so long? So in principle, you would do a case control study. You would take centenarians and you would take regular folks and compare them. And you can certainly do that when you do a GBOS study. However, you cannot do that when you do an methylation study or even a transcriptomic study. And why is that? Well, because chronologic age will confound the relationship, Chronologic age has a huge effect on methylation levels, so of course you cannot compare centenarians to younger subjects. So the best you can then do is you take as cases offspring from centenarians. here, and here we had 63 offspring from centenarians, and we had control subjects who had roughly the similar age, And these data, to go back, come from Claudio Franceschi, Paolo Garagnani, and again several other Italian investigators who secured these families, And incidentally, they also had several supercentenarians or semi-supercentenarians. So the question is the following. Is the epigenetic age of these offspring of centenarians, does that look younger than the epigenetic age of age-matched controls? And that's done here. So in this plot, in the scatter plot on the left-hand side, I show you in orange offspring of centenarians, and in black are just regular folks. And what you see is, as a tendency, the orange dots lie beneath the black dots, and a more formal statistical test suggests that truly epigenetically they're younger than they should be. That's exciting. There is, however, I do want to caution you that this study involved relatively few subjects, and it could well be a false positive. So we won't even publish it until it's validated. But I do want to show it to you more, in certain ways, to stimulate similar studies. Maybe you have access to such data. Yes, a question? Yeah.
How do you control for environmental factors?
Yeah, well, so the question is, how do we control for environmental factors? Well, the controls come from similar regions as the offspring of the centenarians. So I trust my collaborators that they matched subjects, I don't think all of the offspring of centenarians come from one village and all the controls from another village. Then I would be nervous as well, Yes. But let me mention another criticism. Whenever you deal with blood, you should ask the question, well, do differences in blood cell counts relate to these findings? Right? Some people have a lot of T cells, some people CD8 T cells, others have fewer CD8 T cells. That could be a confounder. And so here I show you an analysis of a multivariate model where we regressed the methylation age on centenarian status, chronologic age, gender, and various blood cell counts, just to subtract out the effect of blood cell counts. And sure enough, the result actually becomes more significant, So that's noteworthy. You should ask me one question. How did I come to all of these blood cell counts? Where did I get them from? One way of getting them is certainly to get flow cytometry measures. That would be expensive. But it turns out that the epigenetic clock software, which I will present shortly, actually imputes these data. When you upload whole blood Illumina methylation data, you specify an option which is called advanced analysis in blood, and then it automatically gives you estimates of the abundance of naive CD8 T cells and also what is known as exhausted CD8 T cells and other natural killer cells and so on. Yes?
Can you give me the math behind that?
Say it again?
Can you explain us the math behind that? How it calculates, your software calculates these proportions?
Yeah, so the question is, how does the software estimate the blood cell count measures, And what it does is, for each cell count, there are two different mathematical approaches. For the lower cell counts I mentioned here, CD4T, natural killer cell monocytes, and granulocytes, I use a published algorithm by Hausmann et al. There's a paper in, I want to say, Genome Biology from 2012, where they developed an algorithm. But the other cell counts, the naive CD8 T cells, and the second name is very long, which means CD8 positive, CD28 negative, blah, blah, blah. For these cell count measures, I use my own algorithm that I developed, which is again based on a penalized regression model. So I use certain CPGs that are very accurate predictors of blood cell counts. Yeah. Other questions? Yeah.
Have any standards to use
Whether there's a standard? Or? It's automatic. You upload your data, you get an estimate. That's it, you know. I mean, there's not, you have, so the question is, can you choose parameter values? No, you know. Remember, I actually often don't give too many parameters because I feel the user will hate me for it, you know. But when you know, this software basically has no parameters almost, you know. But I will just explain the software in the second session after we've had a break, you know. Which reminds me, do you want a little break or should I continue? Who needs a break? Nobody, okay. I'll go on for a few more minutes and then we'll take a break, you know. But I just want to come to another application. Some of you study infectious diseases and people have hypothesized that HIV infection accelerates the age of the subjects. Why? Because often HIV-positive subjects present with a higher prevalence, certainly, or incidence of age-related illnesses. And here, of course, I applied the epigenetic clock, and I applied it to, here are brain data, which the finding is quite puzzling. So I had brain samples from HIV-positive subjects and brain samples from control subjects. And what we find, the red dots are HIV-positive subjects. Sure enough, overall, brain regions look older. But when you drilled in deep, it turns out the occipital cortex and the cerebellum showed evidence of age acceleration. However, the frontal lobe did not show evidence of age acceleration. And so I think of this more as a starting point of future validation studies. I'll show it. It's a bit hard to explain. Why does the virus affect the occipital lobe but not the frontal lobe, you know? And here it is.
Individuals had HIV dementia.
No, some of them did, yeah. So the question is HIV dementia related?
Correct.
And yeah, let me mention an unpublished finding from our collaboration with Andy Levine. Sure enough, the people who have HIV dementia, their brains are actually slightly older than the brains of HIV-positive subjects who did not have dementia, you know. A very small study, 54 subjects, but yes, there's a relationship to HIV dementia. So this is in brain, but another question you can ask, what about blood? In blood, I'm very confident in the finding that the blood tissue of HIV-positive subjects is older than that of HIV-negative subjects. Why? Because we observe it in lots of data sets. I really trust that finding. You know. So briefly to mention the software before the break, and before you run away from me, there's an online software where you really give your email address, you upload your data, and then the software will output on the one hand, estimates of cell types, for example, plasma blasts, which are activated B cells, and as I mentioned, the Hausmann estimates of blood cell counts. But also it outputs several measures of age acceleration. There is a measure that, and I need to apologize, it has a very long name, and it has a long name because this is a beta version and we are evaluating these measures. And my software outputs way too many measures and I will throw away over half of them. But just to keep track of things, I gave things a long name. So there's this AAHO adjusted for cell counts. What does that mean? The Horvath epigenetic clock measure adjusted for blood cell counts. That's what it means. And I call this the purest measure of age acceleration. Pure, why? Because it subtracts out blood cell counts. And mathematically, it's defined by regressing methylation age on chronologic age and these blood cell counts and getting a residual, you know. So what does that mean? If you have a blood data set and this measure relates to your phenotype of interest, let it be autism, then you would be able to argue that there's a true epigenetic age acceleration effect independent of changes in blood cell counts, you know, if you have that measure. There's another measure which I call the quote-unquote impure measure of age acceleration that has a very long name, and it is impure because by construction it has a very strong correlation with blood cell counts. It has a strong correlation with the abundance of naive CD8 T cells, for example. And so that measure has the advantage that it actually is strongly associated with all-cause mortality. This measure will literally predict whether you die earlier or younger in a prospective way, and it's prognostic, So I love that about it, but the drawback is that the biologic story of that measure is less exciting. Why? Because it measures in a certain way immunosenescence. It measures, for example, whether the abundance of naive CD8 T cells in your blood is low. And so if you tell a biologist that, well, if you have a low abundance of naive CD8 T cells or a lot of senescent CD8 T cells, then and then you will come down with the disease, the biologists say, oh, come on, that's trivial. Yes, if your immune system doesn't work, it will not detect cancer cells, it will not protect you from infections. Low immunity, sure enough, you will die early, So in this sense, the biologic story is on some level trivial, But in any event, it is certainly a measure that we use. And I want to briefly mention that HA stands for Hannum. So I apologize. There's another epigenetic measure for age, which was described in an article by Greg Hannum and Trey Eidecker and Kang Zhang. And it's basically a measure that they developed, but we modified it. Think of it as a generalization of the Hannum measure. All right, so as I mentioned, this epigenetic clock, actually both measures, predict all-cause mortality later in life. So we have access to several cohort studies where we found that. And I want to mention one article by Riccardo Marioni, Naomi Ray, Ian Deary, Andrea Beccarelli, and many other people who contributed, for example, various cohorts and then showed that this epigenetic clock predicts mortality. And that result was so exciting in Scotland that it made it to front page news in the Scottish newspaper, All right. So I do distinguish, just to wrap things up, I do distinguish two types of measures of biologic age. What I call extrinsic aging versus intrinsic aging. So extrinsic aging is a biomarker in a certain way that is a better predictor of mortality than chronologic age. In certain ways, smoking, for example, we know smoking affects your mortality. You die early. Therefore, smoking will affect extrinsic age, by definition. However, I also have a second interpretation, what I call intrinsic measure of age, and that is simply the intrinsic tissue property that increases as we age, even if you have the perfect lifestyle. What do I mean by that? Imagine there's a person who exercises every day 2 hours. They only eat salad for every meal. Perfect lifestyle. Guess what? They will die, Something in their tissue continues to age, And that's what I call the intrinsic property of aging. And in my opinion, and my hope above all, is that this epigenetic clock measures intrinsic aging more than extrinsic aging, And therefore I say, for example, smoking will clearly affect extrinsic aging, but probably not the intrinsic age of blood tissue, And so in conclusion then, I think many studies that involve telomere length and other biomarkers can really be revisited using epigenetic age to evaluate whether certain stress factors or toxins affect the epigenetic age. There's user-friendly software on my webpage, and in the second session today, I'll review that software for the people who stay on. And for the people who have methylation data, I do mention that don't pre-process your methylation data too much. So some of you may have heard of methods like SWAN, BMIQ, lots of ways of processing methylation data, and these are important and valuable methods. And you should pre-process the data if you do an EWAS study. However, when you measure the epigenetic age using my software, I actually suggest you don't process it too much. Don't remove batch effects and so on, because by doing it, you may throw off the data and induce too many missing data. So thanks for staying with me. I will now stop and we'll do a 15-minute break. Then I'll talk for 30 minutes about the software. But some of you, if you feel like you've had enough, I thank you for attending the workshop. Yeah. Okay. See you in 15 minutes. Yeah. Oh, thanks.